All right, let's go ahead and get started. A couple of quick announcements. So, one, Julia's doing a review session tonight, 4 to 5, and Predlin 306. Come with questions. She's also got some review questions to work on if there's time. The second announcement is I'm going to be putting together between today and Friday a list of times when I'll be available next week and when Julia will be available next week for coming and just drop in and ask questions about things related to this first exam. Okay. So those were the two big announcements. I don't think I have anything else right now. Here's what we're going to do. Break into groups. So what I want you to do is break into a group with those that you're doing you did the voice thread with. So I have the sneaking suspicion that not everybody knows who's in their group, so I'll just kind of tell you where to go. So one is going to be in the back over there. Two will be in the way back, where the row of seats that are all by themselves. We'll do three kind of where, let's see, where Taylor is sitting right now, but kind of like over in this area, and then we'll do 4, 5, 6, and 7 is somewhere in the middle. Sorry, 7. Or wait, there's an 8, isn't there? Yeah. What number of those do we not have? Damn it, I forgot, because something happened. I got to figure that out. Okay, never mind. So again, 1, 2, 3, 4 will meet over in this cluster, 5 here, 6 here, 7, and then 8 will kind of be like over in the middle over here. So one of those numbers does not exist. I don't know which one. You don't need to take everything with you. Just take maybe a notebook, maybe your computer if you want to. What I'm going to have you do is spend, each person gets about 60 to 90 seconds describing their part of the voice thread. So if you had these are the parts of an enzyme, point out those parts, help to describe them. If you're not the one who is presenting, listen. Try to find mistakes if there are any, because there are definitely a few mistakes in there. Okay? All right, go ahead. 1, 2, 3, 4, 5, 6.
You really could have gone on that one. Girls, where are you going? Right here. 6. Right here. Yeah, see? Do you know the number? It's not loading in my,
No, not off the top of my head. Let me check.
We all know what it should be.
Okay. Like the part you did, so like I did. Molly was the first I didn't actually look to see. No, what's the other
1. So like back over there with Allie, Alexia, and Morgan.
You need to stop dropping your I don't get a break.
I'll come join you guys. All right, so what I wanted to do is to take a few minutes to go through this now as a full group, but also to take the time to maybe address any issues that I saw in the individual voice threads. Let's just start with this right here, basic structure of an enzyme. And let's, as we're looking at the structure, let's define the parts as well. So what is a substrate? What binds the enzyme? What's being worked upon, right? Because the function of an enzyme is to take a substrate, turn it into, or take a substrate or substrates, turn them into products. Okay. Where does the substrate bind? Binds to the active site. What else can bind to the active site? Good. Competitive inhibitors combine there as well. So the name implies that between the substrate and that inhibitor, they're both competing for the same site. What else might we find close to the active site? Coenzymes and cofactors. What is in general do both of these do? So what do cofactors and coenzymes in general do for enzyme function? Oftentimes we say that they're required in some ways, okay? And often we will find them in the active site. This image is kind of a classical one of what these enzymes look like, but this isn't in reality what it looks like. It makes it look like the cofactor is a huge part of the enzyme. Cofactors are oftentimes ions, things like zinc, or something we'll see in the next few days, iron. And oftentimes they can be involved in accepting electrons or creating an ionic bond for a short period of time and then breaking it afterwards. So cofactors are going to be required for the full functionality of the enzyme. And then coenzymes are a subtype of cofactor. Again, required for the enzyme function, but coenzymes are organic molecules. One's made up of carbon, nitrogen, oxygen. The main examples that we talk about in here: nicotinamide adenine diphosphate and flavin adenine diphosphate, NAD plus and FAD. And these two are involved in our redox reactions, or we'll find them in dehydrogenase enzymes, reacting with dehydrogenases. So a lot of these different coenzymes and cofactors are going to be involved in receiving electrons or giving away electrons at the end of a reaction. What is this area here? What might we call that? Allosteric site. What is the definition of an allosteric site? Okay, that's one way to describe it, right? An allosteric site is where a non-competitive inhibitor binds. We can't just look at a protein and say or look at an enzyme and say there is indeed the allosteric site. Oftentimes it doesn't have a nice pocket or defined structure like an active site does when we look at enzymes. Allosteric sites are largely defined by other molecules that bind to them. So a protein can have 3 or 4 allosteric sites. And it's not only a location where a non-competitive inhibitor can bind, it's also the location where a non-competitive activator can bind as well. When non-competitive inhibitors bind to allosteric sites, what does this do to the protein or to the enzyme?
It changes the shape of it so the substrate can't bind?
Good. Changes the shape so that the substrate can't bind. Distorts the active site in some way. And there's a couple of ways this could happen. One, it just changes the shape enough so that you can't it's not a good fit anymore. Other examples of what this might look like would be ones, if you think of your enzyme as Pac-Man, I suppose, or Pac-Woman. That's a bonnet, or beret, whatever you want to call it. And here's where our allosteric non-competitive inhibitor binds. What might happen is it might change the shape of the enzyme so that now the active site isn't even open for the substrate to bind. Okay, so sometimes these non-competitive inhibitors can actually almost close up the enzyme, preventing it from even interacting with the substrate. Allosteric activators or Non-competitive activators, on the other hand, do the exact opposite. They make it more favorable for a reaction to take place. So they improve the accessibility of the active site to the substrate, increasing rates of reaction. We actually see both of these in glycolysis. And we'll talk about that in a little bit. Okay. What are we missing from here? Active site, competitive, non-competitive, substrate. I think that's it that we wanted to define. Let's take a look then real quick. Not sure if I've got this in here. 3 types of enzymes. Let's define each of these, and we'll take a look at examples. Examples of each of these actually exist in glycolysis, which we're going to spend the rest of our time with today. What is a kinase? It's an enzyme that does what? Phosphorylates. In other words, it takes a typical reaction we might see would be it takes ATP plus some substrate, and it transfers that phosphate from ATP to the substrate and yields ADP as the other product. These enzymes can work in both directions. Okay, so kinases can create ATP by stealing a phosphate from a substrate or molecule, or it can phosphorylate a substrate by releasing a phosphate from ATP. Obviously, in the left to right direction, that would be energy releasing, because we talk about ATP as being our currency for energy. The other one is going to require some sort of energy to be put in, or requires some energy as well, but usually the energy that's released allows for that phosphate to be added to the ADP to form ATP. This, we would also call, another term for this process is what we call substrate level phosphorylation. As the name would imply, here's our substrate. At the level of the substrate, it is being phosphorylated by ATP, and vice versa. This is how ATP gets generated in glycolysis. Okay? And this is in opposition to what we call oxidative phosphorylation, which we'll cover on Friday. One thing to keep in mind here with kinases and phosphates in this case are always bound to a molecule. They're always being moved from one molecule to another. And we'll make that contrast with oxidative phosphorylation on Friday. What is a dehydrogenase? And quite a few people had a good answer for this one in their responses.
Can you ask your question again?
I can. Thank you, Nancini. What's a dehydrogenase?
Does it take out water?
Not water. Alana? What's that? Hydrogen atoms. Or hydrogen ions. H+, right? Also, a couple other things that we can describe with this one. In biology, these are involved in redox reactions. Redox reactions involve the movement of electrons as well. And in biology, at least in the examples that we talk about, whenever electrons move, hydrogen ions are moving as well. So you can identify these reactions fairly easily by following where electrons go, or more easily by following where hydrogen is moving. Okay? The other thing or other trends that we might look for with this would be NAD+, and its reduced form, NADH. Reduced means that it's gained electrons, and in this case also gained hydrogen. And also FAD, which is the oxidized form of this couple of FAD and FADH2. So those are coenzymes that will be associated with specific dehydrogenases that are involved in moving electrons. And these electrons serve as our high-level energy source for respiration, electron transport chain, and oxidative phosphorylation. Let me give one example of this real quick. So here is an example of a biological redox reaction. In this example here, we have a molecule with 2 electrons, 2 hydrogen ions that are going to be moved onto the NAD. What we would say is that the NAD here is actually the, oops, that's not what I wanted to do. NAD is what we call the oxidizing reagent, or oxidizing substrate. This is the reducing reagent. Its goal, or its purpose, is going to reduce NAD to NADH. And so we would say that in this case, NADH is the reduced product, and it's gone through a reduction reaction, and this orange colored molecule has been oxidized, or it is the oxidized product, and it has gone through a process of oxidation. Okay. And in this process, we're just moving high energy electrons from one atom to another, or from one molecule to another. Last one are the isomerases. What is an isomerase? And in here we've got the word isomer. What are molecular isomers, or molecules that are isomers? What are their features? No, that's not right. Oops. Is that right? Yeah. Here are isomers of each other, I think. What's true about isomers? Alana?
They have
Same chemical formula. So in this case, same, if I've drawn this right, same number of carbons, hydrogens, and oxygens. Different properties, or in this case, it may be a different way to define that would be different shape, because structure defines properties as well. Okay? So isomers, or isomerases, are enzymes that take a molecule and turn it into a different form or a different structure, but retain the same formula. Okay? We'll see this in glycolysis as well. So, for example, switching glucose to fructose, they both have C6H12O6 as their molecular formulas. The shape differs, though. Okay? So the way to identify isomerase reactions, or ones that are catalyzed by isomerases, is just count up the number of atoms, and if they're the same, you've got an isomerization. Okay. What we're going to focus on for the rest of today is looking at glycolysis. I find memorizing glycolysis to be a complete waste of time. I am not asking you to memorize glycolysis, okay? But we're using it as an example to understand how energy is produced in bacterial cells. In bacteria, every single organism that we know of goes through glycolysis to generate energy. What we will find is that after glycolysis, there's a decision point. And And that decision point is based on factors like, is oxygen present in the environment? And organisms at that point can go through the process of fermentation, or they commit themselves to respiration. So what we're going to spend time today doing is looking at glycolysis and using it to help us identify these different types of reactions. All right, so let's just do a couple of these together, and then what I'm going to do is have you guys break into groups and work on this, and then we'll come back towards the end to spend a bit of time talking about it. So for example, first one, glucose does not require energy for its diffusion across the membrane. Oh, this is such a great review question. What is the most likely mode of diffusion? So this is actually going back to something we talked about on Friday. Glucose doesn't require energy, but what's true about glucose? Look at in the first image there, or that top image, it shows you what glucose looks like. What are the properties of glucose? What was that? Okay, carbon, hydrogen, and oxygen, and the molecular formula is C6H12O6. What's true about the bonds there? Polar. Can this pass through the membrane by simple diffusion based on that property? No. So how does it have to pass through the membrane? This one says it doesn't require energy, though. Passive or facilitated transport through some sort of pore would be the most likely mechanism. And that's actually exactly what happens. So when glucose enters into a cell, what we find is that within as soon as it enters, the cell ends up actually capturing glucose. It changes its properties. What's the first step of glycolysis? Take a look at that first step there. What do you observe? What's being added to it? So ATP phosphate's being added to it. What kind of enzyme? Kinase is involved in this step. That enzyme is known as hexokinase. It takes a phosphate, slaps it on a glucose. That now locks glucose into the cell. It's not recognized by the glucose transporter and so can't get back out. This ensures that glucose is locked in the cell and ensures that the concentration of glucose inside the cell is always lower than the concentration outside. And then the last one here is a little bit more challenging. This isn't exactly something we've talked about, so I'll make sure to kind of back up here real quick. The overall reaction of glucose with phosphate to glucose 6-phosphate, it's an endergonic reaction. What do we mean by that term? If a reaction is defined as endergonic, it requires what? Requires energy to be put into the system. Where is the energy coming from for this particular reaction?
The addition of the phosphate group?
From where?
The kinase?
Not the kinase. ATP. Because the ATP, if we look at this, and that's the key point, ATP oftentimes is used as a way to provide energy for what seems to be an endergonic reaction. If we look at it in isolation, glucose plus phosphate, and the PI there refers to inorganic phosphate or just free-floating phosphate. If we take those two and try to bond them together, that takes energy to create that bond. The energy to create that is provided by the breaking of the bond between the last phosphate on ATP and the other phosphates. We call those the high energy bonds. Okay, given that, what I'm going to have you guys do now is kind of work at your own pace with somebody else or on your own, working through these problems. We'll take about 10 minutes to work on this. You may not get through all of it, and that's fine. But we'll come back together towards the end to at least look at a few of these and talk about the rest of the stuff. All right, go ahead. Two half reactions real quick. Oop. All right. So the way that I like to look at these is in terms of energy over time. So for example, if we have ATP and that breaks down to form ADP and phosphate, energy being created or energy being released? Release. It's a high energy bond. In other words, if we were to look at this as the amount of energy, and then this is just the time of the reaction on the x-axis, ATP starts here, takes some energy to get that bond breaking, and then once it breaks, there's this huge release of energy. Okay? And so this from the product, or sorry, from the substrate to the product, this is our change in energy. Okay? That's exergonic. Energy is being released here. If we look at the other side of this reaction, so we have fructose with one phosphate on it, and we need another phosphate to be added to it. So now it's got two on there. Well, the energy of those individually starts here, takes some energy to go into the system, and then here is maybe where the energy of that final product is located. And here is that change. Which of these has a larger magnitude of change? Is the amount of energy released greater than the amount of energy required to create the bond, or vice versa? In other words, is more energy being released from ATP when it's broken down, or is more energy required to create fructose 1,6-bisphosphate? More is released. Just take a look at how large the difference is between what we start with and what we end with. In other words, the amount of energy released here is much greater than the amount of energy that it takes to drive the reaction that ultimately what we're trying to drive in glycolysis. If we take these two and combine them together, then that's known as energy coupling. In other words, we take the energy that's released from this reaction right here, ATP being broken down into ADP and phosphate, and this phosphate and the energy that's used can go into this half of the reaction to ultimately create the product that is intended to be created. Overall, if we were to combine all of this into a single reaction, it's exergonic. We're using the energy from ATP to fuel the endergonic half of this reaction, or the one where we're creating new bonds. Okay? There's an importance to this. This building that's required, it's a little bit of input required in the early stages of glycolysis, but there's a huge payoff. And the payoff is not so much in glycolysis, but in the later steps, particularly in oxidative phosphorylation. Okay? We're about out of time, and I know we have not gone through all of this yet. So, here's what I want you to do. Still do the homework for Friday. It is a conversation about oxidative phosphorylation. But I want you to keep working on this too. Okay? And what I'd like to start with on Friday is not to walk through every step of this, but to start with questions. I have posted up on Blackboard in this particular lecture, all the way at the bottom with the review questions, is this same form and an answer key. Additionally, if you would rather receive the answer key through me talking, It's up on YouTube. It's a 10-minute video of me going through this process, okay? Eliana. So the Friday homework is an individual journal entry where you just have to tell me about oxidative phosphorylation. The prompt's right in there for you to look at. All right, have a good afternoon. I'll see you all tomorrow.